to start today with a bit of a personal confession. I'm someone who really craves stability.
I think most of us do.
Yeah, I like knowing my floor is solid, that the roof isn't going to cave in. I generally prefer my house not to rearrange its own walls while I'm sleeping.
That sounds like a very reasonable requirement for survival.
Yeah.
We tend to view chaos as the enemy of structure.
Exactly. But looking at the research for today's deep dive, I had this moment of, I guess you could call it vertigo, because we're zooming in, way in, past the organs, past the tissues, right down to the cellular level. And the big takeaway is, well, it's pretty jarring. You only exist because your cells are fundamentally unstable.
It's a fantastic paradox, isn't it? Life itself relies on a structure that is constantly trying to tear itself down. If your cells were actually as stable as a building, you'd be dead in minutes.
And that's the headline. Today we are doing a deep dive into microtubules. And before anyone tunes out thinking, okay, microscopic tubes sounds a bit dry, the picture these sources paint is, well, it's violent.
It is.
It's a construction site where the scaffolding builds itself at breakneck speed and then catastrophically collapses just as fast.
And catastrophe isn't even me being dramatic. That is the actual technical term biologists use for what happens to these things. It's not a malfunction, it's a feature.
A feature. So our mission today is to try and understand this restless architecture. We're going to look at how you build a skeleton out of chaos, this whole idea of dynamic instability, and the literal robotic walkers that use these tubes as highways.
It really is one of the most sophisticated engineering systems in nature, and it completely upends how most of us think about structure.
So let's start there with that mental image, because when I hear cellular skeleton or cytoskeleton, my brain just goes to bones. I think of rigid beams.
Like the steel frame of a skyscraper?
Right.
And that's the very first misconception we have to just throw out. If your cells had steel frames, you'd be a statue. You couldn't move, your cells couldn't divide, nothing could adapt.
So it's a better analogy.
The skeleton of a cell is more like, okay, imagine a tent pole that can lengthen or shorten itself on command, or completely vanish and then reappear on the other side of the room in seconds.
So it's modular.
Extremely modular. And to really get how it pulls off this magic trick, we have to look at the building blocks themselves. We need to talk about tubulin.
Right. The sources get pretty technical here. They talk about heterodimers. But honestly, when I looked at the diagrams, this thing looks like a peanut.
That is actually a perfect analogy. A double nut. You got two proteins, alpha-tubulin and beta-tubulin. They are distinct, but they're chemically bonded together very, very tightly.
And that peanut is the basic Lego brick.
That's your brick. Exactly.
Okay, so I have a bucket full of these protein peanuts floating around in the cell. How do I get from a pile of peanuts to a structural tube?
Well, they have this natural stickiness to them, but only in a very specific orientation. They stack head to tail, one after another, to form a long string.
We call that a protofilament.
A protofilament. But think about a single string of beads or peanuts. It's floppy, it breaks easily, it has no real structural integrity.
Sure.
So this is where the real engineering logic kicks in. If you want strength, but you don't want to add a ton of weight, what shape do you build?
You build a cylinder, a tube.
Exactly, a tube.
Yeah.
And the source specifies the geometry here, and it's incredibly precise. You need exactly 13 of these strings, these protofilaments, lined up next to each other in a circle.
13? Why 13? Is there something special about that number?
It's all about the geometry, how the proteins lock together side by side. It creates this slight stagger, kind of spiral seam that makes the tube incredibly stiff for its size. But, and this is the key insight, it leaves the center completely hollow.
Which explains the name microtubule. It's a tiny tube.
Precisely. It's a pipe, not a solid rod. If this were a solid rod made of the same amount of stuff, it'd be heavier and, funnily enough, more prone to bending.
It's the same reason bike frames are hollow tubes and not solid steel bars.
Exactly the same principle. Nature is the ultimate efficient engineer.
Okay, so we've got this stiff, hollow pipe. But here's the thing that really confused me in the reading. If I just dump a bunch of lumber in a field, it doesn't spontaneously assemble into a house. How does the cell know where to put these things? It can't just be random.
No, random would be absolutely fatal. The cell has a dispatch center, a kind of master architect. It's called the centrosome.
The centrosome, and that usually sits right near the nucleus, doesn't it? The center of the city.
Usually, yes. And inside this centrosome, you find these little ring-shaped templates. Literally, they're like starting pads. The source calls them nucleation sites.
A foundation.
The foundation, exactly. And this is where the polarity, the direction comes in. Remember we said the building blocks are like a head and tail peanut.
The alpha and the beta parts.
Right. So the cell always plugs the alpha end into that foundation ring. That means the beta end is always pointing out away from the center.
So it's like a built-in compass. No matter where you are on that tube, you know which way is home, back to the centrosome, and which way is out.
And that directionality is absolutely crucial. Without it, the cell would have no sense of up or down, in or out. It's the entire coordinate system for the whole city.
Okay, so we have a coordinate system. We have these hollow tubes growing out from the center like spokes on a wheel. But this brings us to the part of the research that I found the most stressful, this idea of dynamic instability.
It is the defining feature. And honestly, it's what makes them so cool.
To me, it just sounds like terrible construction. The source says these tubes are constantly growing and then suddenly, without warning, they collapse. They just shrink back to nothing. Why? Why would you build a road just to blow it up 5 minutes later?
Because you have to look at it from the cell's point of view. The cell is blind. It doesn't have eyes to see where a chromosome is or where the cell membrane needs to be. It has to feel its way around.
So the microtubule is like a probe, a feeler.
It's a search party. Think of it like casting a fishing line. You cast it out. That's the growth phase. If you don't get a bite, do you just leave the line sitting there?
No, you reel it in and you cast again in a different direction.
That is exactly what the cell is doing. It's a process we call search and capture. The centrosome shoots out a microtubule. If that tube happens to hit a target, let's say a chromosome, it stabilizes. It locks on.
But if it hits nothing?
The cell cuts its losses. It depolymerizes the tube, reels it in, and uses those same exact bricks to shoot a new tube out in a different direction.
That seems wildly inefficient, doesn't it? Just building and destroying over and over again.
It burns a tremendous amount of energy. But the payoff is adaptability. It allows the cell to completely map its entire internal space in minutes. If the environment changes, the cell can tear down its whole skeleton and rebuild it to fit a new shape almost instantly.
Okay, so let's talk about the mechanics of that reel in phase, because the imagery in the notes was so vivid. It all comes down to something called the GTP cap.
Right. This is the fuel gauge. It's the timer. Explain that for us.
So the tubulin bricks that are just floating around in the cell, they're carrying a little battery pack. It's called GTP. When they click onto the end of the growing tube, they're fresh. They're stable. And this forms what we call the GTP cap at the very tip.
So as long as I'm adding fresh, GTP-powered bricks faster than the old ones fade, the wall stays up.
Precisely. But there's a clock ticking. As soon as that brick is part of the wall, it starts to hydrolyze. It burns that GTP fuel, and it turns into something called GDP.
And the cement dries out. It gets brittle.
That's a great way to put it. So the core of the tube is actually unstable GDP tubulin. It's under a lot of tension. The only thing holding it all together is that fresh cap of GTP tubulin right at the tip.
It's basically the movie Speed. If the bus drops below 50 miles per hour, it explodes. If the growth of the microtubule slows down,
the hydrolysis catches up, you lose that protective fresh cap, and then catastrophe.
And that's the literal word the text uses, catastrophe. It sounds dramatic, but it's accurate. Without that cap holding everything in, the protofilaments, those long strings we talked about, they lose their side-to-side grip. They just peel outward.
The peeling banana image.
Yes. They just curl back and completely fall apart. The tube essentially disintegrates from the tip all the way back.
It's incredible to think that this is happening inside us right now. Millions of times a second, a constant fireworks show of building and collapsing.
And it's that instability that lets us live. I mean, if these were permanent steel beams, a white blood cell couldn't possibly squeeze through a tissue gap to fight an infection. It needs to tear down its skeleton and rebuild it to move.
Which brings us perfectly to movement. Because once these tubes do find a target and they stabilize, they stop being search parties.
And they start being highways, the interstate system of the cell.
And this is my favorite part of the whole thing, the walkers. We aren't talking about things just, sliding down a chute. We are talking about motor proteins that literally walk.
Kinesin and Dynein.
When I looked at the animations, I actually laughed out loud. They literally look like little aliens with 2 feet.
They do. They have 2 heads, which we call feet because, biology. And they move hand over hand or foot over foot right along the microtubule track.
And they're carrying luggage.
Heavy luggage, mitochondria, vesicles filled with neurotransmitters. I mean, relative to their own size, they are hauling massive, massive loads.
How do they know where to go? If I'm a kinesin protein carrying, say, a sack of dopamine, I can't just wander around hoping I find the right exit.
Remember that polarity we talked about? The plus end and the minus end?
The compass.
The compass. These little motors are programmed to read that compass. Kinesin is almost always a plus end directed motor.
Meaning it walks away from the centrosome out to the suburbs.
Exactly. It takes cargo from the center of the cell out toward the membrane. Dynein is the opposite. It's a minus end directed motor. It brings materials, trash, signals from the outside back in.
So you have dedicated outbound and inbound lanes. It's highly organized traffic. And it burns ATP, the cell's energy currency, for every single step it takes.
One molecule of ATP for one step.
Roughly, yeah. So just moving a mitochondrion from your nucleus down to a synapse in your big toe, that is an incredibly expensive journey.
The source gave that specific example, moving a mitochondrion. Why would you need to move the powerhouse of the cell?
Well, think about a neuron. The main cell body is in your spine, but the axon tip could be in your foot. That's a meter away. That tip needs huge amounts of energy to fire signals. If the mitochondrion just sat back in the spine, the foot would go dark.
So Kinesin literally drags the power plant down the highway to where the city needs the lights on.
That's exactly what it does.
That is just, it's unbelievable.
Yeah.
So we have construction, we have traffic, but there's one more event where this entire system goes into complete overdrive. The main event.
Cell division, mitosis.
Right.
This is where the stakes are the absolute highest. If this process goes wrong, you get genetic damage, you get cell death, or you get cancer. The system has to be perfect. And the centrosome, our architect, it basically turns into a general at this point.
It duplicates itself. So now you have two architects.
Yeah.
They move to opposite ends of the cell, and they start shooting out microtubules like crazy to build what we call the spindle.
The spindle. This is the actual machine that physically pulls the DNA apart.
Correct. And the source breaks it down into three very specific types of microtubules that make this happen. And the engineering is just so clever because they each have to solve a different mechanical problem.
Okay, so let's look at the problem. You've got two piles of duplicated DNA in the middle. You need to pull them apart, but you also need to push the cell itself apart to make room.
Right. So first, you have the kinetochore microtubules. These are the grabbers. They shoot out from the poles, and they physically latch on to the chromosomes.
A direct hookup.
A direct link. They attach to a protein complex called the kinetochore. These are the ropes that will eventually shorten and reel in the DNA.
Okay, so that handles the pulling, but you said the cell itself needs to stretch.
That's where the polar microtubules come in. These use a different strategy. They don't touch the DNA at all. They shoot out from opposite sides, and they overlap in the middle of the cell.
They kind of hold hands.
Sort of. And motor proteins sit in that overlap zone and push against them, forcing the two poles of the cell apart. It literally elongates the entire cell.
So the kinetochore ropes pull the DNA in, while the polar ropes are pushing the walls of the room apart.
Yes. But there's a third problem. If you pull really hard on something, what happens to you?
You fall forward. You need to brace yourself against something.
Exactly. You need an anchor. And that's the job of the astral microtubules.
Mm-hmm.
Astral, like a star. These shoot outward, away from all the action, toward the cell membrane behind the centrosome.
They anchor the whole machine to the wall.
They do. They lock the centrosomes in place so the whole spindle doesn't just float around loosely while it's trying to perform this incredibly delicate surgery.
Pushers, pullers, and anchors. It's a complete mechanical system.
And you have to remember, all of this, the grabbing, the pushing, the anchoring, it all relies on that exact same dynamic instability. The microtubules have to be able to grow to find the DNA and then rapidly shorten to pull it. If they were stable, mitosis would just freeze.
Before we wrap up, there was one variation on this whole theme that I found really cool. The swimming gear. Cilia and Flagella.
Yeah, yes. The sperm tail or the little hairs in your lungs that clear out dust and gunk.
The structure there is different, isn't it? It's not just one hollow tube.
I know, it's a very stable 9+2 arrangement, a ring of 9 special double tubes surrounding 2 single ones in the center. It has to be robust to handle all that constant mechanical stress.
And how do they actually move? A tube doesn't just bend on its own.
It uses Dynein, again, our inbound walker. But this time, the Dynein is basically bolted down. It's anchored to one tube, and it tries to walk along the neighboring tube.
But if it's anchored down, it can't actually walk.
Right, so it applies force. But since all the tubes are linked together, they can't slide past each other, so that sliding force gets converted into a bending force.
So the Dynein tries to walk, gets stuck, and its frustration just bends the whole tail.
That is a very colorful way to put it, but yes, physically accurate. And that causes the tail to whip back and forth, propelling the cell.
It's just amazing. We've gone from self-assembling bricks to crashing highways to now swimming tails.
It really covers the entire spectrum of cellular life.
So when we step back and look at all of this, the self-assembly, the chaotic searching, the traffic, the division, what is the big takeaway for you? Because for me, it really changes how I view the cost of life.
That's a great point. It is incredibly expensive, but for me, it changes the very definition of structure. We tend to think structure means rigidity, permanence. We want things to be solid. But biology teaches us that true robustness, the ability to survive 80 years in a constantly changing world, requires constant flux.
So the cell is stable because it's restless.
Exactly. If your microtubules stopped falling apart, if we put a chemical in there to stabilize them permanently, which some chemotherapy drugs like Taxol do, your cells would die. They need to live right on the edge of catastrophe to function.
That's a heavy thought. We're spending a massive amount of energy just to stay on that edge.
It's the price of doing business as a living thing. Adaptability isn't free.
Well, on that note, I just want you to go about your day and try to visualize the billions of little Kinesin walkers inside your legs right now, hauling fuel down to your toes so you can get up and walk to the fridge.
And maybe give them a break if your foot falls asleep. They're working as hard as they can.
Thanks for diving in with us. We'll see you next time.
Goodbye, everyone.